Good morning, everyone. I'm delighted to welcome you all today. Today, we'll be exploring a very important aspect of pharmaceutical manufacturing: quality control procedures in sterile manufacturing and packaging. My name is Dr. S.K. Singh, and I hold a PhD in this field. In this presentation, we'll cover the key procedures that ensure the highest standards of quality in sterile environments. We'll look at the processes involved in maintaining sterility, quality checks, and best practices in both manufacturing and packaging. So, let's get started and dive into the critical world of quality assurance in sterile pharmaceutical production. Let's talk about the purpose of quality control in sterile production. The main goal here is to ensure that the products are sterile, safe, and properly identified, with the correct potency and purity. This is crucial because patients rely on these products being effective and safe for use. Another key aspect is preventing contamination, both microbial and particulate. Contamination can compromise the product's safety and efficacy, so strict controls are necessary to keep the environment and the products free from these risks. Regulatory compliance is also a major focus. We need to meet various standards and regulations to ensure product reliability. These include FDA guidelines like 21 CFR Parts 210 and 211, which cover Current Good Manufacturing Practices, or CGMP. There are also international standards from organizations like ICH, EMA, WHO, and the USB, EP, and IP monographs, which set the benchmarks for quality and safety in sterile manufacturing. Within this framework, we focus on several key areas: ensuring sterility assurance through rigorous testing and procedures, environmental monitoring to track cleanliness, controls during aseptic processing to prevent contamination during production, and thorough validation and documentation to prove that all processes are consistent and compliant. All of these elements work together to maintain the highest quality standards in sterile production, ultimately safeguarding patient health and ensuring the reliability of the products we deliver. Let's take a moment to look at an overview of sterile products. These are specialized medications that require a completely sterile environment to ensure safety and efficacy. They include things like injectables, ophthalmic solutions, and certain infusions. The key point here is that because these products bypass the body's natural barriers, maintaining sterility throughout their manufacturing and handling is absolutely critical. This is what helps protect patients from infections and complications. Connecting this to our overall theme, understanding the unique requirements of sterile products underscores the importance of strict quality control and aseptic techniques in pharmaceutical production. Ensuring these standards are met is essential for patient safety and trust in our healthcare system. Let's talk about quality control in raw material control. The main objectives here are to verify the identity and quality of our active pharmaceutical ingredients, solvents, and excipients. It's essential to make sure we're using the right materials and that they meet our quality standards. Another critical point is to prevent any contamination from entering the sterile zone, which is vital for maintaining product safety and efficacy. When we perform QC tests, we use a variety of methods. For identity tests, techniques like FTIR, HPLC, and UV-Vis are commonly employed to confirm that the raw materials are what they are supposed to be. For purity or assay testing, HPLC and UV are standard methods to determine the concentration and ensure the material's purity level. Water content and residual solvents are also checked, typically using Karl-Fischer titration or GC analysis. These tests help us control moisture levels and ensure no harmful solvents remain. Additionally, microbial limits are assessed, especially pre-sterilization, to make sure microbial contamination is within the limits specified by Pharmacopoeia standards. For example, water for injection, or WFI, must meet strict conductivity and endotoxin standards before it's used in production. This ensures it's safe and suitable for pharmaceutical use. Overall, these tests are essential to maintain quality and safety throughout the manufacturing process. Let's take a closer look at aseptic processing and the controls we implement within the facility. First, the controlled environment is crucial for maintaining product sterility. We designate different grades of clean rooms based on their criticality. Grade A, which includes LAF and RABS, is used for critical zones where the actual aseptic operations happen. This is where the highest level of cleanliness is required. Surrounding that, we have Grade B backgrounds, which support the Grade A areas by providing a clean environment for personnel and equipment. Then, for support areas, we typically have Grade C or D spaces, which are less critical but still maintained to prevent contamination. Moving on to QC controls, quality control is integral to ensuring our environment remains within specified limits. HVAC qualification plays a key role here, especially the HEPA filters that are used to trap airborne particles. We regularly monitor air change rates per hour and pressure differentials to ensure airflow moves correctly from cleaner to less clean areas. Particle counts and viable monitoring are also essential. For example, we perform daily viable particle counts in the Grade A areas to confirm that we stay within the limits set out in Annex 1, which is our guiding standard for aseptic processing. All these measures, environmental controls and rigorous QC testing, work together to safeguard the sterility of our products and ensure compliance with strict industry standards. Now, let's talk about environmental and personnel monitoring and why it really matters in our sterile environments. Essentially, our goal is to ensure that sterile products are produced with zero viable contamination risk. That means maintaining the highest standards to prevent any microbial presence that could compromise product safety. To do this, we perform various QC checks. These include active air sampling, which helps us assess the air quality in real time, settling plates and contact plates, which are used to detect any particles settling onto surfaces, surface swabs, to check for contamination on equipment and work surfaces, and operator gloved finger sampling, which is crucial since personnel are a key part of the environment. Now, when we review these checks, we have acceptance criteria to determine if everything is within safe limits. For example, in Grade A areas, those critical zones, we aim for zero colony-forming units, or CFUs, during critical operations. That means no viable microbial counts should be detected, ensuring the environment remains as sterile as possible. Overall, consistent environmental and personnel monitoring is vital because it helps us verify that our controls are effective and that our sterile products remain safe for the end user. Now, let's talk about in-process controls, or IPCs, during manufacturing. These are the key checks we perform to ensure product quality throughout the process. First, we conduct filter integrity tests both before and after filtration to make sure our filters are functioning properly and not compromised. Next, we monitor temperature and humidity carefully, as these environmental conditions can significantly impact the quality and stability of the product. We also check pH and conductivity regularly, which helps us verify that the chemical properties stay within the specified ranges. Additionally, visual inspections for particulates are crucial to detect any visible contaminants or debris that could affect product safety. It's important to note that the critical IPCs vary depending on the dosage form. Different products have unique requirements and risks, so these controls are tailored accordingly. Overall, these IPCs are essential for maintaining consistent quality and ensuring that each batch meets our strict standards before it moves forward in the process. Now, let's take a closer look at sterilization controls. First, we have the sterilization methods. One common method is filtration, specifically using 0.22 micron sterilizing filters. These filters are critical because they effectively remove bacteria and other contaminants from liquids, ensuring product safety. Another method is autoclaving, also known as steam sterilization, which is typically used for sterilizing equipment and some heat-resistant products. It's a reliable, widely used process that employs high-pressure saturated steam to achieve sterilization. We also sometimes use ionizing radiation, depending on the material and product. This method can be very effective for sterilizing certain pharmaceuticals and medical devices, especially when other methods aren't feasible. Moving on to quality control acceptance, it's essential to verify that these sterilization processes are effective. For filters, we check their integrity through tests like bubble point or pressure decay tests, which confirm that the filters are properly sealed and functioning correctly. Similarly, sterilizer validation and biological indicators, or BIs, are used to confirm that sterilization cycles are effective. BIs contain known spores that are resistant to sterilization. If these spores are inactivated, we can be confident that the process is working as intended. In summary, these controls ensure that our sterilization processes are reliable and effective, maintaining the safety and quality of our products throughout manufacturing. Let's now look at the liquid injection QC, specifically the finished product QC tests. These are critical steps to ensure the safety, efficacy, and quality of our liquid injections. First, we perform a sterility test following USP 71. This is essential to confirm that the product is free from any microbial contamination before it reaches patients. Next, we conduct a bacterial endotoxin test, or BET, which checks for pyrogens that could cause fever or adverse reactions. Ensuring endotoxin levels are within acceptable limits is vital for patient safety. We also perform assay or potency testing, often using HPLC or UV methods, to verify that the active ingredient concentration matches what we intend to deliver. Additionally, particulate matter testing per USP 788 helps us detect any unwanted particles that might compromise the product or cause issues during administration. Lastly, pH and osmolarity checks are performed to confirm the solution's compatibility with the body, ensuring comfort and safety for the patient. For example, a batch of liquid injections must pass the endotoxin limit of less than or equal to 0.2 EU per milliliter, depending on the product standards. This comprehensive testing process helps us maintain high quality and safety standards throughout production. Now let's talk about the powder injection, lyophilized, QC, focusing on the specific quality control parameters we need to monitor. First, content uniformity is crucial. It ensures that each batch has consistent potency, which is vital for patient safety and efficacy. Next, moisture content is measured using Karl-Fischer titration. Controlling moisture is essential because excess moisture can compromise stability and shelf life. Reconstitution performance is another key parameter. It tells us how well the lyophilized powder dissolves and becomes ready for use, which directly impacts administration efficiency. We also perform sterility and endotoxin testing to confirm that our product is free from microbial contamination and endotoxins, safeguarding patient health. Lastly, lyophilization cycle validation is critical. This process validation guarantees that our freeze-drying cycle consistently produces the desired quality, especially in terms of moisture levels. For example, we set a target moisture level, such as less than or equal to X% before packaging, according to the formulation specifications. By rigorously controlling these parameters, we ensure the final product meets our quality standards and performs reliably in clinical settings. This comprehensive QC approach ties back to our overarching goal of delivering safe, effective, and high-quality pharmaceutical products. Now, let's take a closer look at the critical quality tests for eye drops. These tests ensure that the product is safe and effective for use. First, sterility and preservative efficacy are paramount. They confirm that the eye drops are free from microbial contamination and that the preservatives are working as intended. Next, we have clarity and particulate limits, which are essential because any visible particles or cloudiness can cause discomfort or even harm to the eye. Then there's pH and viscosity. Maintaining the correct pH ensures comfort and stability, while the right viscosity helps with easy application and retention on the eye surface. The assay or potency test verifies that the active ingredients are present at the correct levels, ensuring the medication's effectiveness. Lastly, container closure integrity is crucial. This test guarantees that the packaging remains airtight and sterile throughout its shelf life. For example, eye drops must pass USB particulate limits and preservative effectiveness tests, which are fundamental to safeguarding patient health. All these tests come together to ensure we deliver a safe, effective, and high-quality product to patients. Let's take a closer look at packaging material QC. When we talk about packaging components, we're referring to elements like vials, stoppers, seals, ampules, and droppers. Additionally, labels, cartons, and leaflets are also critical parts of the packaging system. Each of these components plays a vital role in ensuring the integrity and safety of the final product. Moving on to the QC focus, the first key point is material identity and compatibility. This means verifying that the materials used are what they are supposed to be and that they won't interact negatively with the product. For example, we need to check for extractables and leachables, which are substances that could migrate from the packaging into the medication. Another important aspect is barrier performance. Packaging must effectively protect the product from moisture and oxygen, which can compromise stability and efficacy. Ensuring these barriers work as intended is essential for maintaining product quality throughout its shelf life. Label content verification is also a critical QC focus. Accurate labeling ensures that patients and healthcare providers have correct information about the product, which is vital for safety and compliance. For example, we might perform a siliconization check on glass vials to ensure proper lubrication, which helps with filling and handling. We also validate inertness to confirm that the glass does not react with the product, maintaining its purity. In summary, packaging material QC is about making sure every component is suitable, compatible, and performs its protective functions effectively, all of which are fundamental to delivering safe and effective medicines. Let's take a closer look at the packaging process, QC, specifically focusing on sterile packaging controls. First, we ensure that our aseptic filling line is properly qualified, which means we validate that the environment and equipment meet strict sterility standards. Next, we perform seal integrity tests to confirm that each container remains securely sealed, preventing contamination. Visual inspection is also critical. We look carefully for any particulates or leaks that could compromise product quality. Additionally, we verify label accuracy and batch traceability to ensure everything is correctly documented and can be traced back if needed. To support these quality controls, we use specific tests such as vacuum decay or pressure hold tests to check the container seals. These are highly sensitive methods to detect any leaks. We also utilize automated visual inspection machines, which efficiently identify and reject any containers with visible defects or particulates. For example, we set a rejection rate for visible particulates based on our specifications, making sure only the best quality products reach our patients. Overall, these controls and tests are vital to maintaining the integrity and safety of our sterile packaging, aligning with our commitment to quality throughout the entire process. When we encounter out-of-specification, or OOS, results, the first step is to immediately hold the batch to prevent any further processing until we understand the issue. Then, we need to confirm that our sampling and testing processes were accurate to rule out any procedural errors. Next, we conduct a thorough root cause analysis, or RCA, to identify what caused the deviation. Based on that, we implement corrective and preventive actions, or CAPA, to address the current problem and prevent it from happening again. Typical causes of OOS deviations include environmental excursions, such as temperature or humidity fluctuations, filter integrity failures, which can compromise product quality, and process deviations, like equipment malfunctions or procedural errors. For example, if we find OOS results in sterility tests, an investigation might reveal an HVAC failure. In that case, our CAPA would involve repairing the HVAC system, requalifying the environment, and retraining staff to ensure proper procedures are followed moving forward. This process is crucial because it helps us maintain product quality and compliance, ensuring safety for patients and integrity of our manufacturing process. Now, let's talk about documentation and best practices, which are crucial for maintaining quality and compliance. First, under documentation essentials, we have several key records to keep in mind. Batch manufacturing records are fundamental. They document every step of the manufacturing process to ensure traceability and consistency. Environmental and personnel monitoring logs are also essential, as they help us track the conditions and personnel involved, ensuring a controlled environment. IPC reports and certificates of analysis, or COA, verify that materials and finished products meet quality specifications. Sterilization validation and filter logs are important for confirming that sterilization processes are effective and properly documented. Lastly, deviations, out of specification or OOS records, and CAPA records are vital for capturing issues and implementing corrective actions to prevent recurrence. Moving on to best practices, adopting a risk-based approach, as outlined in ICH Q9, helps us prioritize our efforts based on potential impact. Regular training and competency checks ensure our team stays up to date with procedures and regulations, fostering a culture of continuous improvement. Using trend analysis and SPC tools allows us to monitor process performance over time, quickly identifying areas for enhancement. And of course, continuous process improvement should be at the heart of everything we do, constantly seeking ways to optimize quality, efficiency, and compliance. All of these practices connect back to our overarching goal, maintaining the highest standards of quality and safety in our manufacturing processes. For further support, please don't hesitate to reach out. We're here to help you implement what you've learned today and answer any questions you might have. Remember, the resources and contacts we've provided are meant to assist you in applying these concepts effectively. And with that, I want to thank you all for your time and attention. It's been a pleasure sharing this information with you, and I hope you found it valuable and inspiring. Thank you again for being part of this presentation.